Hello everybody, welcome to day 4 of our Bioinformatics program here at CPUT Bellville. I am really thrilled to be back with you to return to the fine topic of sequencing. But today, instead of talking about assembly and annotation and stuff like that, we're going to be talking about Amplicon sequencing. Because there are quite a few tasks specific to PCR-based methods that I think have bioinformatic capabilities of their own that we should talk about. So that's today's emphasis. I think this will be a relatively short lecture. I know I've been pounding you for like 2 hours each of these mornings. Not going to be like that today. This is a relatively short deck. But we're going to start by a discussion of PCR primers. I think a lot of people don't necessarily see PCR primer selection as an informatic task. So I'll try to explain some of why that's challenging. From there, we'll move on to talk about measuring microbiomes and metagenomes. These terms kind of get bandied about a lot. If you have these in your grant proposals, then people say, you're cutting edge, and if you don't, maybe you're not being too conventional. So we'll try to talk about what those things represent, particularly in the context of things like 16S and 28S rRNA sequencing. And from there, we'll move on to a discussion of characterizing diversity, because in a lot of cases, the folks doing microbiome research are trying to make the argument that the set of species present in the sample would change in response to whether someone has diabetes or not, or whether someone is a smoker or not, or whatever. So we'll talk about some of the measures that we use for diversity on that score. And then we'll talk about some of the limitations of trying to quantify from evidence of this sort, from 16S rRNA. So I'm hopeful that we can make this seem a little more quantitative to you and not just kind of a hand-waving area of research. It's certainly not. So there are many competing goals that we're trying to accomplish in making PCR primers. So just to remind ourselves, our ability to amplify, to make many copies of a region, requires that we have a primer in the forward and reverse directions that bracket this area that we want to clone out, basically, that we want to amplify out of a mixture. So those primers have the particularities of those sequences have many different goals to meet. So first off, we want to employ primers that melt at the same temperatures. So remember that these primers are going to hybridize with the DNA, and if you want to allow them to lift away from the be removed from the DNA, you want to raise the temperature enough that they come off together, that the forward and reverse primers can pull away together. If you have one primer that's very GC rich and another one that's not, they're going to have different melting temperatures as a result. Target-specific primers, obviously this is a very clear requirement. If you have 15 places in the genome where a particular primer can lie down, that's not very specific. You want the primer's specificity to be such that this location and this location only is where this is going to sit down. You want to define efficient primers. If your primers are not allowing you to double the amount of DNA you have in each cycle of PCR, that's a real limitation. So finding ones that are very effective, very, very efficient for this process, that's great. Don't inhibit Taq DNA polymerase. Obviously, if we're using the Thermus aquaticus enzyme for synthesizing the DNA, it's going to be a problem if we're preventing it from functioning by using sequences that stick to it. So that's not great. And one of the most challenging things that comes about is the problem of homology among among the primary sequences that we use. So what does that look like? So in this case, the visualization we're using comes from this Untitled et al. Paper. You can see that the white primers here represent the forward primers, and the black ones represent reverse primers. So you could imagine a variety of different kinds of primer-primer interactions. You could, if you have a bad primer design, find that your forward primers interact with themselves. if the primers are all lost to the PCR reaction because they're interacting with themselves, that's obviously no good. So we see that the forward could interact with the forward, the back could correspond to the back, or you might have this weird interaction where the forward and reverse primers anneal to each other. Not very viable. If we look at letter C here, we're looking at interactions where we don't have these full body interactions between the primers, but rather kind of an end effect. So we need to be cognizant of the fact that we may not have a whole primer-to-primer interaction, but rather one that just sort of affects the tails. They're both really damaging to our ability to use the primers. There's also the ability to form hairpins. I think everyone knows that at least messenger RNA can form all kinds of interesting hairpins. A tRNA, for example, is an example of this complex stem loop formation, right? So hitting stuff like that in your primer is really problematic because if the primer is so busy sticking to itself, it's not going to anneal to your target DNA. And of course, the more regions that you're trying to amplify out of the DNA, the more interactions are possible. If you have, for example, 5 different targets that you're trying to amplify all at the same time, how many different interactions can you have among just the forward primers? Right? So you have 5 forward primers. You could have A interacting with A interacting with B, A interacting with C. All of these are possible, right? So that kind of multiple interaction becomes much more problematic as we rise to a higher number of sites to amplify. Now, I would note that I'm not doing an in-person walkthrough on the software today, but in prior years I've done some online videos at the YouTube channel that talk about how these tools can work. I believe I walked through Oligo 7 in one of those videos. So feel free to look those up. But Oligo 7 is software published way back in 1989. Not Oligo 7, that was, I think, the first edition. But it's been a perennial favorite, and they keep adding new features to it over time. So that's been quite lovely. But it's also true that you can get information on improved primer design through the Primer Web software. This is built into the NCBI Primer Blast to tell you about which interactions are present with that. So you can find that at this URL. That was published back in the year 2000. But again, to assume that software has been holding still since its original publication is almost always a mistake. In fact, if you find that there's a bioinformatics tool you want to use, but you notice that it hasn't been changing in the last 5 years, it's quite likely that something has superseded it and that it's no longer under active development. It's always worth looking into what's replaced it, if that's the case. So I hope that the video for Oligo 7 can help illustrate some of the features we can deduce about our primers in that video. Okay, so from here we're going to talk about microbiomes. My first exposure to microbiomes was not a clinical exposure. It was not an example of that sort. It was the question of acid mine drainage. Have people heard of AMD? One of the effects that we get from mining of various sorts is that we very frequently get very low pH water pools generated. You've driven through Joburg at some point? You maybe have seen these huge hills of tailings, right? A lot of times the environment's interaction to having these great heaps of awfulness and so on can be that we end up with anomalous pH pools. So in the United States, I was working with someone named Jill Banting who was working with acid mine drainage, very acidified water, which had relatively small numbers of bacteria interacting within them. And so our question was, which bacteria are present in what combination? And how does the interaction among multiple species go beyond what you could anticipate from just a single organism? All organisms work in combination with other organisms, even humans, right? I mean, me and my house cat do a lot more damage to the environment than I would by myself. My cat's a killer, I just got to say. She's violent, terrible temper on my cat. She's brought home her 5th mouse in 2020, just like 2 nights ago, right? So we have interactions with other species, so do bacteria. And if you only study bacteria in isolation, you don't really understand how they can function as part of a bacterial community. So microbiome research really moves us in the direction of understanding how ecologies function when multiple players are at work. Okay. Today, of course, when you talk about microbiome, you are very, very frequently talking about clinical research, trying to understand the microbial communities of the gut, for example, or of the urogenital tracts, or for that matter, what grows in our lungs when we breathe the air, right? So we certainly can have fungal communities, for example, living in our lungs. It's not a great condition, but it happens. So I want to note then that the microbiome is not just about bacteria. The microbiome is about microbes, and microbes can be from multiple kingdoms, right? So for bacteria, our emphasis is almost always on 16S rRNA. Now, I've talked a lot about messenger RNA just 2 days ago. I think we're all familiar with that. I have already mentioned tRNA, transfer RNA, which brings the amino acids to the ribosomes. But what about this rRNA? Does everyone remember what that's about? It's ribosomal, right? So one of the things that I, as a protein guy, was a little surprised by when I was a postdoc is that I had thought of ribosomes as protein machines, that these proteins are going to connect another amino acid to this growing chain of polypeptide that's here. But in reality, that's kind of a mistake. In a lot of respects, a ribosome is a protein structure that contains within it biologically active ribosomal RNAs. That in a lot of ways, the rRNAs are doing the work of ensuring that everything is in the right position for amino acids to be connected together. So while I tend to think of ribosomes as these proteinaceous machines, without their ribosomal RNA, they're not doing much. Okay? So a ribosomal RNA then is one of these molecules within the ribosome that is responsible for this activity. So why is it that we study the 16S rRNA? Does anyone remember what 16S is all about? Svedbergs. How large a subunit this is as it migrates through, oh gosh, is this a gradient or is this through a gel? I've forgotten. Gradient, gel? Okay, well.
No, I was thinking is the length of the sequence, the sequence of that.
Right, so generally speaking, Svedberg is a unit for how something migrates through another. So yeah, the longer the sequence, the larger the bulk, the bigger the subunit size, basically. But in this case, I think that this is about movement through a gel, if I'm not mistaken. All right, so this 16S ribosomal RNA is this genetic sequence that we are going to target in order to understand which bacteria are present. So I think that's a really important first point. If you're going after bacteria, 16S rRNA is a good option. If you're going after fungi, it's not your friend, right? Fungi have a very different ribosomal RNA sequence that can be used in this way. In both cases, though, what we're looking for is this very special combination of highly conserved regions that serve as the locations where the primers can sit. And they need to be flanking a highly variable region, one that changes quite a lot from one species to another. Does everyone understand why? I'll talk about it for a minute then. Okay. So if you're going to make a primer that is useful across a wide variety of species, you need to target a region that's highly conserved across taxonomic space. Because if you make a primer that works for these genera but completely not for those genera, you're going to get a pretty good interrogation of these, but next to no interrogation of these. Your amplification will be blind to that. So when we target the 16S rRNA, what we're interested in getting the sequence from are these V regions. You see those red ones marked V1, V2, V3? The font's going to be really small from there, but the red bits in this top sequence. Those variable sequences are the ones that help us to resolve which species is present. But we can't put a primer in that space because it's so variable. To make a primer that matches to that variable region will give us the ability to resolve things that have the parts that aren't on the primer, but that very primer will not be applicable to species that have a different sequence at that location. So we then think of creating forward primers, here shown as green arrows, sitting down in constant regions, and reverse primers that are sitting down in constant regions. But we want them to be flanking a V1 sequence, or flanking a V2 sequence, or flanking a V3 sequence. How is it going to alter matters if we're using a sequencer that produces long reads rather than short ones?
Sorry, come again.
How will it affect our primer design if we're using a sequencer that generates long reads rather than short ones? This is not a one-size-fits-all operation. If you have a sequencer that's producing reads that are only 40 nucleotides long, your forward and reverse primers need to be really close together, really. If you are able to sequence out a longer region, like PacBios or whatever, you have a little more distance allowable on these segments. And of course, it's quite likely that you're going to be using multiple pairs of primers, that you're going to be creating template DNA for sequencing that covers the V1 region and the V3 region and maybe V4 or something like that. So we have to be cognizant of the fact that how we set up our primers and how what type of instrument we're going to use for the sequencing can give us different amounts of information from which we're going to deduce which species are present. And which primers you use will also have quite an impact on which types of species are differentiable. Okay, now, I note that 16S rRNA is not the only thing that could be sequenced to give us this resolution. In bacteria, some folks have looked at CPN60. This is a chaperonin. Everyone knows about chaperonins? Proteins that help others to fold properly. Yeah, so the chaperonin 60 protein has a pretty good mix of constant and variable regions to help us understand which species we've got. Now in fungi, we frequently will work with 18S rRNA or 28S rRNA. Those are both viable. And yet, when people have done large-scale microbiomes in fungi, they've frequently found that they don't get enough information from 18S rRNA and 28S rRNA to tell them which fungi are present. So for this, we see that some of them have turned to using the Internal Transcribed Spacer sequence. I've included a diagram here from, I believe this is from a 2016 paper. So they are using the ITS regions as variable regions that are very useful for determining which specific species of fungus is present. Okay, so I hope that this little discussion has helped us to think about some of the biology that we're trying to get at. We need constant regions flanking because we want primers to stick in the first place, and we want variable regions between them that give us enough variation for us to pick out which species this bug is. Yes?
So, like I was told that if you are choosing primers that maybe is below between 16, like that from that end,
yeah.
If you amplify it, then it's going to be your bands, the bands are going to be maybe down, you might not even see.
That if you set your primers in this region,
yeah, that region, yeah. maybe between 68 and yeah.
68 and 136?
Or maybe 68 to 100, or if you want something, yeah, you are going to see the band on the, yeah, when you amplify, yeah. They said it's cool, I don't know, I'm asking if they said it's cool, you said it come out from between 3 something to 5.
Okay.
Yeah,
So producing a longer segment,
yeah,
having a little more space between the primers gives you a better amplification. A better amplification or a better ability to retain the fragment you've produced?
No, to see, I'm talking about the bands now when you amplify. Yeah, the gels, yeah.
This becomes sequence, this is sequencer.
Right, generally speaking, you're not going to attempt to infer the species by how the band migrates on the gel. You expect the length of the thing to be roughly the same across species.
Yeah.
But yeah, if you're just trying to make sure that the amplification worked and you're doing it by gel, if it's too small a fragment, you're not going to see much. Thank you for that. I don't do a lot of hands-on gel work with PCR. It's been years since I did a PCR at all, actually. So I hope I'm not misstating things, but I'm trying to talk about this as an informatic challenge. Deciding which bits you want to amplify is going to depend a lot on what species you're trying to differentiate. Okay. So there is this definition that shows up very, very frequently in microbiome research that everybody should be able to get their heads wrapped around, because without it, this field becomes a bit of a mystery in a hurry. And that is the Operational Taxonomic Unit, OTU. Generally speaking, you could make the mistake of thinking that if you see a one-off sequence, if you see a particular sequence produced from this amplification, that every different sequence represents a different species. You could be under that misconception. What that's going to lead to is that every time you have a sequencing variation, like a missed base call in that amplified region, you're going to invoke yet another species to explain it. And that's not very parsimonious at all, because after all, lots of bacteria have lots of sequence variation within species. I mean, bacteria grow really fast, right? And they adapt to match their environments very rapidly. So you could have an E. coli from my guts, or an E. coli from that's been a lab strain for years, and you would find all kinds of sequence variation that differentiates those two populations. So, generally speaking, we do not treat each sequence that differs from another as another species. In almost every case, we're going to use clustering to allow us to say what are the different clouds of sequences. We have a whole bunch of sequences that are like this with at least 97% similarity. We have this other cloud of sequences that are within 97% similarity of each other, and so on. And we call these different clouds OTUs. So generally speaking, when we try to say which species are present, we are not trying to hang a label of species on every sequence. We're trying to put the label of species on these clouds of similar sequences. So you might ask, where is this 97% coming from? Is this a magic number? It's shown in red, that makes it authoritative, right? In fact, it's not very much so. A lot of this is what we call a rule of thumb, and you don't want to have to rely on it too much. If you see two sequences that are 96% similar, they may not be clustered together because of that disparity, but that doesn't mean you have another species. It just means you have kind of this gray zone where things are very similar to this cluster of sequences, but not as similar as we would expect to lump them in as OTUs, as the same OTU. Okay, so generally speaking, we find this whole set of sequences that are quite similar to each other. We form a consensus sequence, consensus meaning something that captures kind of like the most typical sequence through that OTU. And then we will try to figure out from that consensus sequence what the best genus and species label to hang on that OTU would be. We'll be talking about different software that we use for that purpose. But getting this notion right away that we are clustering together similar sequences and calling them individual OTUs is a really important first step. So it may be that the sequence differences you observe are natural sequence variation, that all the different E. coli bugs in my guts have very slightly different sequences, and so this is biological variation within the species that we're capturing in this cloud. However, we are of course using massively parallel sequencers, and there are so many sequences that these things generate that we have a fair number of sequencing errors to draw from. We don't want to go chasing after sequence errors to no purpose. So anticipating that both biological and technical variation are producing this variety of sequences is an important step as well. That's why we have this loosey-goosey 97% similarity rule. So we then, I hesitated to put this last statement up here. Each OTU is a different species. Well, that's if I could be Luke Skywalker for a moment, I would say almost every part of that is wrong. But I would say that in practice, we think of the set of OTUs as representing the species that are in that sample. I hope that's not too fuzzy, that we have some number of species. We don't have direct access to that, but we do have the sequences that we've derived from them. We judge a set of OTUs based on those sequences, and we hope that those are representative of what species were actually there. Okay, now, just because you have a sequence that has resulted from your PCR amplification of 16SR RNA does not mean, and maybe you have a whole bunch of them, a whole bunch of sequences that are all in this cluster of related sequences. It may yet be that we cannot pin down a genus and species. Now, why is that? The answer is that at some point we need to have a lookup to say, if I have an E. coli and I've sequenced out variable region 1, the sequence I should see is blah, blah, blah. Right? That's our lookup table. What if the sequence that I have for variable region 1 does not match any of the sequences in my database that allow me to look up the species name? It might be that I can say, well, this is of order blah, 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 or class blah, 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 or if I'm lucky, genus blah, blah, blah. But I may not be able to get all the way down to species name. So our ability to recognize that an OTU exists is not the same thing as our ability to hang a species label on it. Okay, great. So you can find a lot of information about OTUs in that paper. I'm trying to include those for later lookup if I'm totally botching the description. A lot of these concepts, I think, can benefit from a look from different perspectives. So reading the papers that help to substantiate these points is valuable. All right, so I'm going to highlight 3 tools for this purpose. These are not exhaustive by any stretch. Microbiome research is popular and hot, and there's not a year that goes by where a new software toolkit of this sort was published. I made these slides about 3 years ago, and already profoundly out of date. So feel free to add others that you think should be on the list. So we start with MOTHUR. Now, if you look in the upper left corner, you see the logo for that. I have to say, the people at MOTHUR have a very good graphic designer working on their logo. I feel safe using this software. Isn't that pretty? That's really lovely. Okay, so this was published in 2009. I think it's the Schloss et al. Paper. It scaled, one of the things that they were really focused on doing is making sure that people could use next-gen sequencers, massively parallel sequencers, for the purpose of microbiome research. Obviously, if you limit yourself to only being able to do 384 templates in a run, that's going to hold you back from getting a lot of depth in sequencing. So they wanted to create software that was scalable up to millions of reads. They succeeded. They wanted to use kind of a histogrammic approach that if you are looking at a particular, say, V1 region or V3 regions, that you could ask, how popular is each of the sequences that I've produced in this particular variable region? So what would that look like? Would you expect that every sequence you see for a particular variable region would be distributed evenly from the most common one to the least common one? Typically not. So we've already mentioned two reasons why you're going to see a large number of one-off sequences: biological variability and sequencing variability. So you can imagine a long tail then of many, many, many sequences that are seen only once. There are going to be some sequences that we see very, very frequently. If you're looking at Dave Tabb's E. coli guts, well, you're going to see a whole bunch of E. coli variable region sequences. Great. So this histogram of how many times each of the distinct sequences is observed is valuable for that purpose. It handles clustering of OTUs. So this is one of the first tools out there that showed how valuable clustering was in the context of next-gen sequencing with microbiomes. And at different distance thresholds. Now that term might not seem to make a lot of sense right away, but this ties right back to that big red 97% in the earlier slide. That we weren't sure at first that 97% variability in these variable region sequences was a reasonable place to draw the line for an OTU. So MOTHUR tries a whole bunch of different ones. What happens to your clustering if you allow for 95% similarity? How much better or worse does it get if you move to 99% similarity? So MOTHUR allows you to do all of these things. And it's one of these first tools out there that does a really good job of reporting diversity measures, something that we're going to talk about soon. Okay, now I look at this second software, and I mean, at first I thought, why are they calling it QIIME? But then I decided it was more like maybe a Chinese sh sound. So can we call it QIIME? What would you call it?
Isn't it QIIME?
Here we have a vote for QIIME.
But it's pretty QIIME.
I thought it was QIIME, but then QIIME is there's also software called QIIME for doing protein structures, so I don't want to just lump them all in by mistake. All right, well, I'm going to call it QIIME, but we can call it QIIME if we like. All right. So one of the nice things about QIIME was that their emphasis was not on use our software or go away, but rather creating a framework that we have different approaches we can use for computing similarity. Pick the one you want. We have different methods available for handling the clustering. Pick the one you want. We have different methods for doing visualization. So QIIME is a framework. It's a modular framework allowing us to substitute out the various pieces. Here we've got their logo up here. All right, so that's really powerful. A lot of approaches now in bioinformatics have worked on scalability and modularity, the ability to substitute out this particular piece if it doesn't work the way you want it to. A lot of people, for example, have used the Galaxy framework or K9 or something like that. These are workflow managers that allow you to stitch together related tools. QIIME is an example of that in the context of OTU estimation. Now this UPARSE algorithm I thought was really a curious one when I read through its paper. UPARSE appears to be, I would say that the author gives the air that people who've done this before didn't know much about software engineering. Let us show you the right way to do it. That's always a dangerous atmosphere to leave your readers with. Because frankly, when you send a paper to a journal and you have that kind of attitude, the first person who receives your manuscript for peer review is the person you just trash talked, right? But UPARSE has a lot of really good ideas in it, and it seems to be really carefully created. They, however, have really lousy graphic artists. See that? Now, if you had the option of having that icon on your desktop or that, where are you going to go, right? I'm just saying. Okay. People make decisions about software for worse reasons than that, trust me. Okay. So, UPARSE begins by saying, if I see a sequence only once, it could not possibly be valuable. Chuck it all out. You could be throwing out 50% of your data set because all of those individual V1 sequences were seen just once in one read. That's kind of a sensible thing to do. Think how much simpler the problem of OTU estimation becomes if you can throw those out. And look at this, filtering chimeras during clustering. Do people know the word chimera? A chimera, well, in the old days, would be a mythical beast that combines multiple animals within it. So the Sphinx, right? Human face, body of a cat, right? That's cat. How about a centaur? Body of a man with the backside of a horse. My father occasionally called me the backside of a horse, so I mean, maybe I'm a centaur, right? I'm just kidding. All right, Dad would laugh at the joke. It's okay. So we could imagine then that you might, through this weird PCR reaction, have produced a sequence that combines the V1 back half of one species and the V1 front half of another species. So that might seem like, so what's the big deal? That's just a weird thing and it happens. But when you're trying to cluster together sequences, chimeras like that can cause this OTU and that OTU get stuck together erroneously. So being able to filter out chimeras is actually quite powerful. So I'm not going to tell you that you need to use MOTHUR because it has a beautiful icon, or that you should use QIIME because it's a modular framework, or UPARSE because this arrogant person was actually right on. You know, there are most frequently the software that you choose for assessing OTUs out of your sequences is going to depend on whether there's a postdoc in the lab who's done this before. What did they use? That, in effect, is how these decisions typically get used. But because new software is generated all the time for OTU estimation, it's worth your time to think about which one is most applicable to the research question you are asking. All right, so these are brilliant tools just to get to a place where you've got your list of representative consensus sequences for each OTU in hand. Maybe you have hundreds of these things. Maybe you have thousands of these things, right? At the end, we still need to be able to say, what does this sequence, putatively from V3 of 16S rRNA, what does this sequence show us is present in this mixture? And for that, you'd better have a lookup table. So we're going to talk about the Ribosomal Database Project, originally published in 1997. When I made this slide a couple years back, they already had 3.3 million different 16S rRNA sequences, each with some sort of taxonomic information attached to it. They had 126,000 28S rRNA sequences. So these are useful for bacteria. These are useful for fungi. All right. So RDP, you can see their logo down here, is the ribosomal database. I've seen a lot of people using GreenGenes. It was originally published in 2006, and their effort really drew attention to why we needed to remove chimeric sequences from these databases. That if you leave them in, a chimeric sequence can match to 2 different OTUs, and it's only got taxonomic information correct for one of those. So that's not a big deal. SILVA is another very commonly used one from the ARB toolkit. There's a little icon for it. And here they're trying to provide information about large and small subunit sequences. So which of these would be valuable if you were going after fungi? Are you going to go after one that says the 16S rRNA toolkit? Probably not. That's bacterial. Now, does that mean GreenGenes has nothing in fungi? It doesn't mean that. It may be that although this is their icon, over the last 2 years, maybe they've really built up their 28S collection. Maybe they have the ITS sequences as well. People have found that if you try to go after fungi with just 28S, you're losing a lot of ability to resolve different species after all. So each of these sites has been working on building out what kinds of species they best represent. And probably with every year that passes, there's another repository that's making its claim to fame in this space. So I would ask you to think of this identification process as being one in which we are going to get a consensus sequence to represent each OTU. That's what the prior slide was all about. But when we want to translate that into taxonomic information, to actually say, oh, this is Escherichia something, this is Bacillus something. This requires that we're able to translate these consensus sequences for a variable region into a taxonomic identity. It's almost always done through lookup. Now we have a couple other approaches that, as people get more and more ambitious in what they want to accomplish out of these, out of a complex community of bacteria or a complex community of microbes in general, is that people are moving to much more open-ended data collection practices. So metagenomics is an approach that there was the old brutality in war that people would talk about, kill them all and let God sort them out. That's a grotesque way to operate. But in fact, a lot of people in metagenomics kind of make the assumption that you can sequence everything and untangle at a later date which bits of the sequence you've produced came from this species rather than that, right? This is a really complex, challenging bioinformatics approach. So if you're able to release DNA from all the nuclei of all the bugs in a bucket of seawater, for example, you now have a mixture of DNA that represents all these different species. You can shiver all of that up, submit it to sequencing, and get back a patch of sequence from here and from here and from here with all these different bacteria or all these different fungi contributing bits of DNA to the overall scenario. So what do you do with that? Any notion what you do with that? Okay, let us assume, this is just an assumption, let's assume that some of the species in your bucket of seawater are sequenced. They've been sequenced in the past individually, they've been grown on agar plates, we know we know their chromosome if they're a bacterium, or we know their chromosomes if they are fungi. And now, when we look at the BAM file, the FASTQ file that's been produced by the sequencer, we can look at where all of these reads map to species we believe to be present. Right? Now, maybe you're really, really aggressive and you have downloaded all of GenBank, something awful like that, and you're now trying to map all of these reads to all of GenBank. Okay, that's a bad scenario, we don't want to do that. But mapping is a relatively fast exercise, and you can map all of these reads to all of these genomes that exist, that are known to exist. However, I would note that a great many of the bacteria that we find in nature have never been cultured, let alone annotated. Lots of things just don't live well in culture. So, if you're depending on the ability to grow colonies of this stuff on agar as the first step to doing sequencing of these things, you're already going to lose a lot of the bacterial diversity in nature.
Only 1% is culture.
Only 1% is culture. Well, yeah, that's a pretty small cut. All right. So I wanted to suggest the possibility of the second half of what we talked about on Monday, gene assembly. It is possible to do a mapping to major players in the sample and maybe account for 20% of your data, possibly. That's a very big number. But let's say some of it maps to E. coli and some of it maps to Bacillus subtilis and Staph aureus and blah, blah, blah. Big, well-characterized, very common bacteria. We can then say these reads have all been accounted for. Let's look at everything else. So now we have the other 80% or other 50% of our reads that we still need to account for. we can do a de novo assembly from those to say, well, these sequences all overlap by all but one nucleotide. We can stitch them together. And now we have the ability to say, this long stretch of DNA, this contig, represents the reads. Now we need to figure out what species that's from, or what functionality that encodes. That's metagenomics. What if instead of starting with genomic DNA, we start with messenger RNA? How is that going to alter the picture that comes out? When we do assembly, de novo assembly, we're not going to get these long contigs out, will we? But we might be able to rebuild transcripts, complete transcripts from these critters. How will a metatranscriptome differ from a metagenome in the histograms that come out of this, and how frequently we see each sequence? I would note that our bodies transcribe a lot more of some genes than of others. And they tend to transcribe those things that we're making lots of. So if you're curious to know ribosomal sequences from transcription, you're in luck because we generate piles of RNA transcripts. So a metagenome is going to tell us about overall DNA from all the species that are present. The metatranscriptome is going to tell us about what they're doing right now. What biological challenges are they going after? What are they trying to transcribe to respond to whatever is happening? So if you see that a whole bunch of genes in iron-sulfur clusters are being transcribed right now, there's some suggestion that chelation is a big factor in how these bacteria are growing. So what are these bacteria capable of is kind of the genomic question. What genes can these species draw upon to take on their biological challenges? What are they currently doing is much more responsive to this question of metatranscriptomes. Today's lecture. Thank you to Diplomix for letting us have this opportunity to work together. This has been a really fun week, and I'm looking forward to seeing you tomorrow as well. Let us return to our slides. We've talked quite a lot about just the question of identifying which species are present in a mixture. That is not, strictly speaking, the only thing that we care about in doing a microbiome pile of research. And evaluating diversity of species and quantifying species has some kind of complex challenges associated with it. First off, let's start with what many people think of as the primary output from a microbiome experiment. You have a spreadsheet, right? You've run MOTHUR, you've put the files through GreenGenes, and now you have a spreadsheet. Each row of that spreadsheet has some sequence that was spotted for a particular variable region, and it has some putative taxonomic identity to it. For folks who've never done a microbiome experiment before, what fraction of all of the OTUs that you produce in a given experiment will be resolved down to the gene level? Sorry, down to the species level. All of them? Most of them?
I'm guessing, because you're saying like something that's similar to, so maybe the percent that's...
Yeah, it's going to depend upon how well characterized that microbiome has been in the past. If you're working with soil bacteria, you may find that some of the medically oriented databases are going to be less well covered. So in fact, having produced a set of OTUs, and having created a set of consensus sequences, you may find that which database you pass it against, RDP or GreenGenes or SILVA, gives you a very different recognition rate. And it may in fact be that the same consensus sequence gets perceived as two different species depending on which database you throw it against. In any case, you should never expect that every consensus sequence will be unambiguously resolved down to a particular species. This is just not going to happen. So we have a lot of unknowns left to us in the microbiome space. That said, let's talk a little bit about diversity. Now, species diversity is something that has a nice long history to it from the world of ecology. And generally speaking, you'll find that diversity gets separated out to 3 different levels when we do ecological diversity. So we have alpha diversity, which talks about within samples of a single cohort. Right? So maybe you have a stream site where you're sampling out what species are found here, and if you took several different plugs of soil from that same riverbank, you're going to get different species each time, but they're going to overlap in some way that tells you what that stream bank has on offer in species diversity. When we go to evaluate things like what is the species diversity of riverbanks before we pass into this forest or after we pass out of this forest, or what it is upstream of the industrial polluters and downstream of the industrial polluters. I'm sorry, I'm showing my anti-corporate bias again. Then you're likely to see differences between cohorts, upstream and downstream, for example. So we call that beta diversity. More generally, if you're asking what is the total diversity seen in all sites associated with this particular watershed, now you're getting into something more like gamma diversity. But let's talk about microbiomes in the medical context. If I have 5 people who don't have diabetes and 5 people who do have diabetes, is it reasonable to say that the alpha diversity allows us to compare among people in the same cohort? Not really, because just because you and I don't have diabetes, or you do, maybe you don't have, but among the people in the room who don't have diabetes, I would note that what I had for breakfast for the last 40 years and what you've had for breakfast for the last 20 years, youngsters, is going to have a lot to say with what my microbiome looks like today. And there's a lot more determining what the species diversity of my guts are than just that, right? So generally speaking, when we talk about alpha diversity as being samples within the same cohort, the only way you could really deal with that is to say, I've got multiple samples drawn from the same site for the same person. Right? So generally, as soon as you step from one person to another, you've already moved into beta diversity when we get into microbiomes. So you're going to want to, before you go publishing about alpha and beta diversity in a particular organ system or a particular context, I would strongly urge you to see where they set their bounds on what alpha diversity versus beta diversity is. Generally speaking, as soon as you are comparing among different people, beta diversity is already in play when we're talking about their internal organ system microbiomes. Okay? Great. You can learn more about these terms in the 2 papers that I've listed here at the bottom. This paper from 2012 in Nature by the Human Microbiome Project really does help to set a lot of useful context for how to talk about medical microbiome biodiversity. So we have next generation sequencers, we have massively parallel sequencers that are producing volumes and volumes of sequencing data from individual samples. Does that mean that we have sequences from every species in a sample? It does not, because there's a lot of dynamic range associated with microbial communities, some that are present in really huge numbers and some that are present in really small numbers. So a natural question should be, well, next-gen sequencing has obviously been around for a long time. Is everyone familiar with an Illumina MiSeq? Illumina MiSeq? Have people heard of these? Okay, it's a really, really popular kind of desktop model sequencer from a few years back. But I would stress that at this point in time, an Illumina MiSeq is not considered a contemporary instrument. It's really been surpassed by later ones. So, if 5 years ago you ran a MySeq on your microbiome sample and you had all of the sequences data produced from it, maybe at that time you thought, my gosh, this new instrument is so fantastic, I must have a comprehensive sampling. Just the same today, maybe you're sending your samples over to CPGR for sequencing on the Illumina NextSeq 500. Well, I hate to break it to you, but today, the NextSeq 500 is not considered like the top-end sequencer for world research, right? So the data that you produce today are always going to look shoddier and less comprehensive in 5 years than you would like. And that's because biotechnology is always on the move. Our abilities in sequencing in the course of my career are astonishing. I would never have believed that massively parallel sequencing could ever work when I was in grad school. And now we take it as a foregone conclusion. So, anytime you have a data set, it is worth asking the question, how comprehensively have I sampled the species in this mix? Even if you were able to, even if you have like the next generation sequencer at your beck and call, I promise you in 5 years, you're going to think, well, we could do that better now. So, rarefaction represents one of the ways that we ask, how completely have we sampled this mixture? And the way it works is a bit confusing, I think, for people. It comes from the term rarefy, to take a random subset of a given size from the original sample. So if you produced 100 million reads, you might ask, would my conclusion about which species are present change if I were to subsample this, if I were to grab out 50% of the reads drawn randomly, and I recreate my OTU list? Do I get the same number of OTUs, or does it shrink a lot? This rarefaction procedure helps us to get a sense of how comprehensively we've done this. So you could, from your 100 million reads that you've produced in an experiment, you could do a 50% draw from that 1,000 times over, and for those 1,000 experiments, see how many OTUs are represented by the data set. And if those numbers dance around a lot, that's worth being cautious about. It suggests that which subset of the reads you pull is going to have a huge effect on the OTUs. If you have a truly comprehensive sequencing collection, that shouldn't happen. You should be getting roughly the same OTUs back in each of those cases. So they're used for understanding the depth of sampling of a community compared with its total diversity. Is this only relevant to microbiomes and sequencers? It's not. So in proteomics, we frequently bemoan the fact that we don't see all the proteins, or that we don't have quite as much sensitivity as is possible with something like RNA-seq, for example. So one might apply rarefaction analysis in that context to say, if I chose 50% of these spectra randomly and went through the identification process, would my protein counts change dramatically? And generally speaking, yeah, they drop quite a lot. So this rarefaction analysis is one of the ways that we use to evaluate how completely we've done this sampling. Yes, question?
You say when you use it in the chapter with depth coverage when you want to look at coverage of your proteome.
Yes, depth of coverage is sensitivity in that context, yeah. So if I am subsampling a proteomic data set, I am asking how much my IDs change if I grab some subset of the PSMs for making that judgment. Ideally, the number of proteins is not going to drop a whole lot, but the sequence diversity, the peptides that we see, probably will fall quite a lot. Good question. So I'd like to walk through 3 rather different case studies in Amplicon quantification. So what if I am studying the course, the time course, of a particular person's tuberculosis case? Maybe they arrived at the clinic a year ago with the first sign of tuberculosis, And because their case has been a challenging one, we've decided to go back and analyze sputum samples that have been collected in a space of maybe every month from that time. This person's been a very willing research subject. Most people wouldn't put up with that. Okay, so now I have 12 months of time for this person. How long is the normal course of antibiotics to cure tuberculosis? 6 months. 6 months. So 6 months from the time that person presented at the clinic, they should have been all clear. But this person wasn't. We found that at first, they had a diminishing TB burden, that they were breathing a little easier, that we had a diminished amount of culturable bacteria in the sputum, etc. But then, we saw that their tuberculosis got worse again. And over time, we realized that this was because their tuberculosis has become drug resistant, or multiply drug resistant. So now I have these 12 sputum samples, and I want to evaluate not which species are present, but what fraction of the tuberculosis bugs growing in this person's lungs are drug resistant. Right? So this is a little different than the microbiome question typically. We're now asking about kind of a subspecies question. It's not subspecies, I'm sorry, that's an incorrect statement. But we're looking for what fraction of the bugs have developed developed drug resistance with time. So this is very similar to the 16S rRNA problem, isn't it? Because we have a clear vision of just a very few genes that enable M. tuberculosis to escape from the standard treatment regimen. So we can, if we're able to pull out and amplify even 10 spots in the MTB genome, we're going to get a sense of what fraction of the reads we get at each of these locations have developed the mutation versus those that haven't. Okay, so that is our first scenario. That's not a typical microbiome, but it's still a very clinically relevant one, isn't it? Okay, next. I spent a lot of time working with people in the Early Detection Research Network, the National Cancer Institute. So I always find a cancer example is easy to come by. So in a tumor is the fraction of key genes containing mutations changing over time. Do you guys work with cancer much? Some do, some don't. Okay, so cancer is a disease of mutation. One of the scariest things somebody ever told me at a cancer meeting was that everyone in this room has had cancer. I thought that was a wild thing.
That is a lie.
Our immune systems are looking for cells that grow without bound all the time, and they're shutting that stuff down pretty aggressively. And when you're young and healthy, you're home free, right? But once you're in, say, your 70s, is your immune system is able to kick the butt of cells that are growing without bound as well? It's not. So in a lot of respects, cancer is the disease that gets you if nothing else does. Sorry, that's a very maudlin thing to think, perhaps, but but that's one of the ways that we think about our world. So I would suggest then that when we monitor a tumor, understanding which mutations they've picked up is important. But one of the key features of cancer is that it is a disease that encourages the formation of new mutations. Like what if mismatch repair is turned off in the cells of a tumor? Now DNA mismatches don't get fixed and mutations linger into the next splitting of the cells. Right? So that's a very frightening thing. Now, are all mutations that can be accumulated in the course of cancer equally dangerous? No, definitely not. In the case of something like mismatch repair, a driver mutation, one that takes place in the genes for mismatch repair, is a much more dangerous thing in allowing for the promotion of the disease later on. If you have a contact inhibition gene, this is one of the mechanisms that our cells use to say, okay, I've grown to touch my neighbors, I should not grow anymore. If you turn off systems like that, growing without bound becomes possible. Metastasis, the ability to cut a cell loose from its environment and go exploring via the circulatory system or the lymphatic system, metastasis is a bad thing, right? So these are what we think of as driver mutations, the mutations in cells that cause this disease to enter a new stage of awfulness. It's a very approximate suggestion. My apologies to all cancer researchers everywhere. But if you are able to monitor a tumor over time, that can be very valuable. And one of my favorite examples to use in this case is prostate cancer. Prostate cancer is one that we think of as grandpa's problem, right? So if grandpa can't pee over the space of a few days, he's going to finally admit to grandma he's not feeling okay. And she might be able to convince him to go to the hospital. I'm speaking for how my grandparents always dealt with doctors, which was not at all. All right, so lots of times we find that we detect an abnormal PSA signal for an elderly gentleman, and we say to ourselves, oh no, grandpa has prostate cancer. But in fact, there are lots of things that can make a high PSA signal happen that have nothing to do with cancer. It might just be a swollen prostate. Prostatitis can also cause a false positive here, a high PSA signal, even though cancer isn't even involved. But even if grandpa does have prostate cancer, there's a tendency on the part of young people to say, gosh, if grandpa has cancer, we've got to get that thing out. So if grandpa has prostate cancer, we're going to send him for surgery. But in fact, we overtreat prostate cancer a lot. And there are real side effects to going through the prostate cancer surgery that men don't like to speak about very much. So, what if grandpa has prostate cancer, but it's an indolent one? It's one that has lots of mutations that have absolutely nothing to do with this disease getting worse. And grandpa's going to live another 20 years, and maybe that tumor will have grown another 10%, but it's not what's going to carry him away. So, being able to monitor the development of mutations within a subset of genes, again, looks a lot like these microbiome analyses, except in this case, we've annotated all the genes. All right. Now, do species change in dominance within this bacterial community as a function of pH? We've talked about the acid mine drainage program that I was part of already, so I'm not going to hit this again. But there's some really fascinating papers that came from Jill Banting's group as part of this work. So looking in natural communities, looking at soil bacteria is actually very relevant. Has anyone heard of Superfund? The Superfund was created to deal with the fact that American industry and research had created some very, very toxic sites. Does anyone know Oak Ridge, Tennessee? Okay, Oak Ridge, Tennessee is where I did my postdoc. Oak Ridge National Laboratories was one of 2 cities built specifically to create materials for the first atom bombs. There was no city there, and then there was a city, because the federal government convinced a whole bunch of researchers and a whole bunch of workers to move to this isolated valley in eastern Tennessee, because if a nuclear accident took place, the hills around it would help shelter the surrounding community. Weird, huh? All right, so places like that have developed a lot of heavy metals in the groundwater. And we're very cautious about, if you've got a bunch of radioactive waste stored someplace, like say Nevada, that is not getting anywhere out, right? So I mentioned this in the fairly innocuous scenario of pH in the soil, but what if it's heavy metal contamination? What if it's nuclear waste storage? You care about whether those things are affecting the microbial communities surrounding it. Okay, so I hope that this helps to broaden us out to think about a much broader scenario where looking for sequence changes in dominance come into play. I kind of wish that I had given the full story on calibration curves as part of this lecture. It's something that I talk about in some other contexts, but I want to talk about kind of problems at the end of this scale. So here we have a sigmoid curve, right? So we have some amount of read count. This is our readout to say how much of something. Generally speaking, if you have many more members of an OTU present, you expect the number of reads associated with them to rise. Everyone feels comfortable with that. However, it might be that people assume that if any amount of that bacterium is present, you're going to get some number of reads from it. And that's not true. It is also possible to generate reads that coincidentally match a sequence, even though none of that species is present. So we have this problem of detection at the lower level. We can't really be sure that a singleton read actually represents a truly identified species. And this problem down here at limit of detection is a really big one. Now, level of quantitation is usually to say that we have some range in which the variability of measurement is so low that we can accurately quantify this species. All right, so maybe if a bacterium is present at least 1% of the community, we can detect that. And if it's up to 30% of the community, we can measure that pretty well. But we also have this problem of attenuation at the high level, that the number of reads doesn't really grow if you have, say, 50% of the population being a species versus 40%. I don't know where those limits are. So we have problems of detecting something that's very rare and of quantifying something when it's present at a really high level, because it's basically blasting out everything else at that point. Both of these problems are things that we need to be thinking about. Read count is not something to be evaluated simplistically. Just because you have 5 times more reads in one sample than another is not a reason to say there are exactly 5 times as many examples of this bacterium here than there. The relationship is not linear when you get to extreme values.
So, like, what do you think we should do in this case?
Well, the best thing to do is to be able to measure it. For example, if you have a sample that's run twice, you're able to determine for these species that are in the same relationship in both samples, does it appear to be at the same level? Do you see that the numbers, that the variability associated with a 5% member of the population is plus or minus 20%? Or is it plus or minus 2%? That technical variation is a really important thing to be able to capture. You may also have batch effects and stuff like that that can interfere. So a lot of the topics we discussed in gene expression show up here as well. How else could our counts mislead us? Different target sequences may have different primer efficiencies. If you're trying to get at a V1 region and you're using a nearly constant region but not completely constant region for your primers, you may find that you are omitting or diminishing the counts for a particular species because of that oddity in the sequences. So the primer design is certainly a big issue when you're trying to determine quantity. Sequencing errors may cause us to believe that species exist in our samples that don't. So the problem of eliminating singletons, or at least rare reads, is something that can help us to resist falsely claiming a species is there. And stochastic noise. Stochastic is a very handy word. If you have random bounces in your data, we frequently call those stochastic noise, especially in counting scenarios like this. So in the example of running technical replicates of the same sample, if you observe the same sample in 2 different sequencing experiments, and you see that the numbers are moving, even though the same sample is the same between the 2, that's an example of stochastic noise. So it may be that just random effects cause you to believe a species is changing in quantity, even though it isn't. So we have to be cautious against all of these things. So quite a few takeaway messages. Bioinformatics tools support a lot of key activities when we're dealing with Amplicon sequencing. You should have some sort of tool that's ensuring that the primers you select are not incompatible with each other. You should use clustering of reads to group together those that had very, very similar sequences. And generally, we're going to expect that something that's within 3% similarity, 97% similarity, 3% dissimilarity of each other come together to be an OTU. We will then annotate these clusters of sequences with taxonomic information. Hopefully, the database we're working with has good information for the types of bacteria that interest us. Determining how completely your sequencing has sampled the organisms that are present in a mixture is very important. Being able to quantify remains a very difficult challenge, and having some notion of technical reproducibility is going to help us quite a lot in that case.